A-Level Biology 9700, Paper 4, Variant 1, October, November 2024, question number 7 and 8. So we will start from question number 7. And question number 7 is, Spea multiplicata is one of the several species of American spade-foot toad. Young spade-foot toads are called tadpoles and live in water in ponds. So S. multiplicata tadpoles show three different phenotypes due to genetic variations. The three phenotypes are detritus feeder, intermediate, and carnivore. Detritus feeders are small and carnivores are large. Intermediates vary in size between the two extremes. A detritus feeder and a carnivore are shown in figure 7.1, and three types of uh, tadpoles are there. Detritus feeders eat detritus, small pieces of dead organic material and algae, and have smooth mouth parts, small jaw, muscles, and long intestines. And intermediates can eat all available food and have teeth-like mouth parts, medium-sized jaw muscles, and medium-sized intestines. And carnivores eat fairy shrimps and other small animals, have teeth-like mouth parts, large jaw muscles, and short intestines. Now we will move towards the question. And question is, in pond 1, they observed in pond 2 ponds 1 and 2. So first of all, the pond 1, the scientists observed a high density tadpole. High density tadpole means, it means the population was high. High population, and high population, know leads towards the intense competition. Intense competition for resources, and due to this intense competition for resources, the resources are scarce or less availability, less available, less available. And the third point is, the most of the tadpoles they counted were either detritus feeders or carnivores with very few intermediate present, only detritus plus carnivores. So these two types of tadpoles were available and very few intermediate were present. It means these two were leading in, and the population of these two was greater. So question is, describe and suggest explanations for the type of natural selection that appears to be acting in pond 1. So it means two types of tadpoles are there. This shows a type of natural selection that is called as disruptive selection. This is the type of selection, disruptive. This disruptive selection. And what is disruptive selection? So disruptive selection is a type of selection, natural selection, where individuals with extreme traits, individuals with extreme traits have high fitness. Now question arises, what does it mean that individuals with extreme trait? Extreme traits means their titles, they only depend on organic material. They only eat the organic material. Carnivores only depend on the other animals because they are carnivorous in nature. Detritus, which are eating only the organic material available in the pond, and there is no intermediate or very few intermediates which you can say negligible type of number is over there. So we can say detritus and carnivore, only two extremes. They are only detritus, they are only eating organic material. Carnivores are only eating the organic, only depending on the other animals. So it means there is no intermediate who are eating each and everything. As for example, the intermediate tadpoles are very few not available or very few available. So it means these two extremes are there. So these two extremes shows the disruptive type of natural selection in which these two types of extremes are present. And due to these two, you can say extreme type of uh, population or organisms or the tadpoles with which are with intermediate feeding strategies are less efficient because both detritus feeding and carnivory are higher in number. So this results a bimodal distribution of feeding types, a high number of detritus feeders and a high number of carnivores with relatively few in between. So which are in between, there will be very few. So in this way, we can say that there will be the why they are acting because uh, these uh, these two types of uh, population or these type of tadpoles they have specialized adaptations in their bodies. Detritus feeders might have special mouth parts. They have might have these special mouth parts, and these special mouth parts can be used for scraping and filtering detritus. And on the other hand, the carnivores have adaptations for catching and consuming prey. So in this way, they have created special types of adaptation. And when natural selection takes place, only those organisms survive which have adapted according to the changed environment. And the other organisms which do not follow or which are unable to adapt themselves according to the change in environment, they are, you can say, they are best fit in that environment. And so the nature select only the best fit organisms. So here in this case, the detritus feeders and the, you can say, the carnivores, they will best fit for the environment. So this type of natural selection is called as disruptive type of natural selection. Now I have written this question. After this explanation, you can read this question. The observation described in Pod 1 points to disruptive selection, which is also known as dicertifying selection. So, disruptive selection in Pod 1 favors tadpole with specialized feeding strategies such as detritus feeders or carnivores due to intense competition. So, for food, leading to the elimination of intermediate feeder. Intermediate feeders, they uh, were not having these special adaptations to the environment because they were depending on all type of food. So detritus, they were eating the organic food, and because the number was very high. On the other hand, the other type of organisms, they were depending on the other organisms, so they were eating up these intermediate organisms as well. So in this way, we can say only these two types of organisms which were greater in number, they survived in that environment, and due to their survival, the intermediate feeders, they were best fit in that environment, due to which they were eliminated from that environment, or very few were left. So in this way, this uh, type of selection was only uh, favored these two types of disruptive and carnivorous organisms. Now we will move towards the next question. And next question is, in pond 2, the scientists observed A low density tadpoles. Low density tadpoles means indicates the less competition for resources. Low density, less competition. Sufficient food availability for all tadpoles. Less population, less competition for food, so the availability of food was ample. Food was available. Opposite to the pond one. The most of the tadpoles they counted were intermediates, leads to the best fit environment for intermediates. Survived. So describe and suggest explanations for the type of natural selection that appears to be acting in pond 2. So in the scenario of these 3 points, that is the low density of tadpoles, sufficient food availability for all tadpoles, that most of the tadpoles they counted were intermediate. Now, instead of extremist, detritus or which depend on organic material, carnivores only depend on other organisms or the animals. There were more availability of the intermediates, which were depending on type of food. So this type of natural selection is called a stabilizing selection. This type of selection is stabilizing selection. This type of and stabilizing selection, also known as purifying selection, is a form of natural selection that favors individuals with intermediate traits while selecting against individuals with extreme type of organisms are eliminated or very few in number as comparison to the organisms which are intermediate, which depend on all type of food items or the food resources available to them. So in a stable or so in this way, stabilizing selection, a stable environment is produced. Stable, stable environment is produced because there is high density, low density of tadpoles. Food resources are available in greater number, so only they will survive. So optimal, we can say that the average phenotype is often the most efficient and utilizing those resources. Tadpoles with intermediate feeding strategies must be generalist, able to exploit a wider range of food resources because they are depending on all type of food. So they will explore the all type of food resources. So there will be a reduced risk of you can say of the carnivorous tadpoles which depend on other animals. So in this way, um, the carnivores risk is reduced. So predation is also you can say also reduced. So in this way, if predations are present, tadpoles with extreme traits might be more conspicuous or vulnerable. So we can say that opposite to pond one, in pond one, two types of tadpoles were available over there because they were best fit in that environment in pond one, where there was the high density of tadpoles. But here there is low density of tadpoles, the only organisms which are depending on all type of foods, or we can say they were the intermediates which are depending on all types of foods they are available in this because there was sufficient food availability and they had no risk to be preyed by the other type of organisms, which especially the carnivores, detritus, feeding animals, they are also not available, or they are very few number in these. So these are in these intermediates. So in this scenario, there will be the opposite conditions, because in this case, nature has selected the organisms which have the qualities to eat or to depend all type of food items. So scenario in point 2 suggests stabilizing. So stable environment is there. So this is called as stabilizing selection. Stability is there. So the stabilizing selection in 4-2 favors the average phenotype intermediate, which offers a balance of resource utilization, reduced risk in a stable environment with low competition and abundant food. If abundant food is abundant, competition will be less, because food availability is more as comparison to the number of populations which are present in that pond, due to which the competition is also reduced, and due to reduced competition, the availability or the survival of the species which are present will be increased. So in this way, both of these ponds will show the different types of natural selection behavior. Now we are moving towards the next question. The intestine length of S. multiplicata tadpoles show continuous variations. Continuous variations. Continuous variations means traits that vary gradually over a range of values. So sketch a curve on figure 7.2 to show how intestine length varies in the tadpole populations in pond 2. Uh, Here I would like to say that the type of stabilizing selection is there. And you know, the stabilizing selection favors the average phenotype, which is the intermediate intense length in this case, and select against extreme variations. Continuous with type of variations are there, and it can take an average of values, not just distinct categories. So average value will be peakest value. So the here in this case, when we sketch the curve, it will be just like this. So peak in the middle, it shows the intermediates. These were two extreme. They can be detritus or they can be carnivores. So the peak of the curve is in the middle of the axis. So it is representing the average intestine length. This signifies that the highest number of tadpoles have this intermediate length. So the we can see the both sides. The both sides are tapering. So these sides indicating a decreased number of tadpoles with intestine length that deviates from the average. Average is in biggest number, in highest number, and the other two types, detritus has longest intestine and carnivores had very short. So intermediates are surviving in this type of stabilizing natural selection. So it will start on the axis, and we can see uh, this will look like a classic bell-shaped curve. Bell-shaped. So in this bell-shaped curve hai, and then we can see this is showing the stabilizing selection into the pond 2. Next question is, a student suggested the variations in S multiplicator tadpoles could lead to sympatric speciation in some populations. outline the features of sympatric speciation. So first of all, what is sympatric speciation? So sympatric speciation is the evolution of new species. Evolution of new species from a single ancestral species while both continue to inhabit the same geographic region. So this is the definition of sympatric speciation. Now we have to outline the features of sympatric speciation. First of all, the first feature of this is it occurs within the same geographic area. Occurs within the same geographic area. So sympatric occurs within the same geographic area. So it means uh, unlike the other types of speciation, as you know, the allotropic speciation, where populations are physically separated. But here in sympatric speciation, when new species evolve from a single ancestral species, uh, while they are living in the same location. They are living in the same location, but the new species are originating over there. In the same geographical area, the new species are originating. So this is the type of, or this is the first feature of sympatric speciation. They are at the same geographical area and new species are originating from it. And the second, we can say the reproductive isolation develops within the population. So the second feature, we can write that reproductive isolation. Isolation occurs within the population. So we can say in this type of evolution, the mechanism of this evolution, it prevents the gene flow between the diverging groups within the population. This can happen through different forms, polyploidy, you know, a rapid form of speciation, and the habitat differentiation. So populations may begin to use different habitats or resources within the same area, leading to adaptations that reduce interbreeding. You know, when they adapt, or when they you can say occupy the different habitats due to differentiation in habitats, the other resources within the same area. So it will also leading to adaptations to reduce interbreeding, so they will not interbreed with one another, so the adaptations to that area will be increased. And then sexual selection can also occur, which is, you can say, changes in mate preferences can lead to reproductive isolation. So there will be, due to this, you can say the reproductive isolation can take place between them. As you know, if they prefer the different types of partners for breeding and the different times for the breeding process while they are living in the same area. So other we can write over here, that is the disruptive selection can also play a role of disruptive selection. So it can also play There are different, you can know the selective pressures within the same environment. It can favor individuals with extreme phenotypes. So it will potentially leading to the divergence of the population. And other feature can also be the genetic differences can accumulate uh, genetic differences. Accumulation. So one more I would like to speak, speciation will be rapid. Speciation will be rapid. So these are the different, you can say, the features as, for example, the variations in the tadpoles as in the intestine length as previously we have discussed that could lead to sympatric speciation. So different groups of tadpoles might be evolving, uh, occur within the same, they are living at our same geographical area, or reproduction isolation can take. They are living at a place, they are living in the same geographical area, but reproductive isolation can take place due to in which the different, you can say, factors will affect this type. So this implies that even within the same pond, different groups of tadpoles might be evolving reproductive barriers due to the factors like habitat speciation. Different, they will adopt the different feeding habits due to availability of different types of food resources. So they also prefer the different types of breeding uh, partners, and you can say they will have the different methods or the different time for breeding. So in this way they will adapt all these different types of features in them. So it's important to note that demonstrating sympatric speciation in the wild can be challenging, as it requires careful analysis of genetic and ecological data to rule out the possibility of geographic barriers. So in this way, the sympatric speciation have the different features as allopatric type of speciation. So again, I would like to, the populations, they are living, new populations arises, new species arises, species occur. So when speciation occur, within the same geographical area, so the different factors can act different due to different factors effect, as for example their reproductive isolation. Isolation means they are preferring the different types of partners, they are preferring the different time for breeding or reproduction. And here the disruptive type of selection also play a role. You can say the due to all these factors, the genetic differences are accumulated between those different types of species. So the speciation rate will also be very, will be increased, it will become very fast. So in this way I would like to say the different features can affect the sympatric speciation. Now I will read the answer which I have written after this explanation. Occur within the same geographical area. Done. Sympatric speciation happen when new species evolve from the single ancestral species while living in the same location. They are living at the same time at the same place, but the new species are originated. So reproduction isolation will develop between them. They will prefer the different partners. They will prefer the different piece of time. So within the population, disruptive selection can play a role. Disruptive selection can play a role. It can favor individuals with extreme phenotypes. Already discussed in the previous question, what is the disruptive type of um, natural selection? So potentially leading to the divergence of the population, there will be the change in the features of that population. And lastly, we can, I would like to say that the the rate of speciation will be very rapid or fast. So it is all about this question. Now we are moving towards the next question. And next question is figure 7.3. Figure 7.3 shows the evolutionary relationship between three species of American spadefoot toad. And here you can see on screen that is Sp multiplicata and Sp hominoide and Sp bombifrons. So these different types show the different areas in which they are existing. So I would like to explain how analysis of DNA allowed the evolutionary relationships shown in figure 7.3 to be determined. Here we will explain how the analysis of DNA allowed the evolutionary relationships. So first of all, the first step in this figure, we can see the relationship, evolutionary relationship, which has been shown in this figure 7.3. The first point in this regard is number one is the DNA sequencing and comparison. So analysis of DNA. For analysis of DNA, first of all, there is need of the sequencing and comparison. Sequencing of DNA and comparison. So for this purpose, for sequencing and comparison, there is need of extracting the DNA. DNA should be extracted. So scientists extract DNA samples from each of the three toad species. These three toad species, Apia multiplicata, Hermondile, and bombifrons. So they extract the first step in this is a extracting DNA, and B step in this sequencing for DNA is you can see sequencing. They will sequencing. So DNA sequence determining the exact order of nucleotide basis, adenine, guanine, cytosine, thymine, all this will take place, and then their alignments. So this is the first step. In this regard, they will, and secondly, they will analyze the differences. Second part is analyzing sometimes when we write on screen, some problems can occur. So the second step is analyzing sequences difference. Analyzing sequence differences. So what are these analyzing? So accumulation of mutation, whether you can say mutations are accumulated in DNA sequence, and due to the mutation, the DNA sequence will be different. So longer two species have been separated, the more different will be present in their DNA. So calculating genetic distance. So scientists calculate the genetic distance between the species based on the number of differences in their DNA sequence, and then using molecular clocks. They will use the molecular clocks to find out the differences between the sequence of DNA. And the third, you can see the constructing phylogenetic trees. So these steps can be, and for this third type phylogenetic analysis, they will use the different software programs to use the genetic distance data to construct phylogenetic trees. So clustering based on similarities, and then species within more similar DNA sequence are grouped closer together. You know, the rules of classification. So same rules will be adopted over here. The closely related DNA will be kept into one group. So routing in the out groups, they will be kept separately. So then interpreting the trees. So here, how analysis? So the analysis of DNA will be based on these points. And as I have told you, the first point, sequences of DNA and comparison. For this, they will extract the DNA, they will check the sequence, their alignment, and then they will analyze the sequence, and then they will try to find out the differences which are produced due to the mutations, or and then the polygenetic trees. So they will discuss their, they will make or they construct the polygenetic trees, and then they will interpret the trees. So in this way, it will help them to analyze the DNA. So in this way, they can find out the relationship among different types of DNA sequence. So answer which I have written, you can see over here. DNA analysis allows scientists to quantify the genetic differences between species. That is, with by adopting these four different steps, they are able to analyze or to quantify the genetic differences between species, the close which the DNA sequence, which are very resembling with one another, they will be closely related, which are then used to reconstruct their evolutionary history and create phylogenetic trees like the one in figure 7.3. And you can see this type of phylogenetic tree here, according to this tree, you can see this provide a powerful tool for understanding evolutionary relationships and the timing of speciation events. You can see timing has been also given. So tree in figure 7.3 shows the spe, spe bombiferon and spe hemonide are closely related. You can see they are closely related, and you can see these spe and multiplicata, they are very far away from the multiplicata, and their ancestors lived in about 20 million years ago. You can say their time range is 20 years, whereas spe multiplicata, they originate 40 million years ago. So in this way, by constructing these different types of phylogenetic trees, we can find out the date, we can guess the date and the time of the origination of these different types of species. So these four steps are helpful to find out the exact or to quantify the genetic differences between the species. Now, question number 8. Scientists use many different techniques in genetic engineering. Sometimes they gene for genetic engineering cannot be extracted from the donor organism cannot be extracted from donor organism. Instead, the gene is synthesized using one of the two different methods. Now, come to the question. For genetic engineering cannot be extracted. Sometimes we are unable to extract. We are unable to extract the uh, and then the gene is synthesized using one of the two different methods. So we have to uh, write the two different methods for synthesizing a gene for use in genetic. So here in this case, you can see gene synthesis. Uh, Two types of, I would like to say, two methods can be helpful in this regard. And those two methods can be, we would move towards over here. The two methods, one of them is chemical synthesis of DNA, which involves chemical synthesis of DNA. First method is number one, chemical synthesis of DNA. Chemical synthesis of DNA means it involves building the gene, particularly using DNA synthesizers. One step in this regard is gene artificially using DNA synthesizers. The first step. And the second step is short DNA sequences. In this method, short DNA synthesizers. Sequences. So, the first method is chemical synthesis of DNA. So, chemical synthesis of DNA can take place by using, you can say, DNA synthesizer, which make the artificial DNA. So, in this regard, the short DNA sequence can be used. So, this method is useful when the exact nucleotide sequence of the gene is known. If the exact method is there, if the exact, you can say, then we can see this method can be used. And second method is reverse transcription from messenger RNA. So in this method, the gene is synthesized by isolating messenger RNA from cells expressing the desired gene. Extraction, isolation. First step in this case is A, isolation of messenger RNA from cells. Expressing the desired genes. Type of genes you want to make or to synthesize. Reverse transcriptase enzyme. messenger RNA is used to make complementary DNA strand. So then amplification. So these are the two methods by which the scientists can make the by making use of these two types, they can. So there are two methods. One of them is chemical synthesis of DNA, which involves building the gene artificially using DNA synthesizer. I've written over here. Short DNA sequences are chemically synthesized and then assembled into full-length gene. So in this way, the second method is reverse transcription. Transcription and translation, this is the natural method which are used to manufacture or to synthesize the protein or the DNA molecule. So here there will be the reverse transcription. That is, in transcription, there is the formation of messenger RNA from template strand of DNA. But here it will be opposite, or it will be reverse, as there will be the formation of DNA strand from messenger RNA. So in this way, reverse transcription will take place, and then as a result of this reverse transcription, transcriptase enzyme will be used. And as a result of this, there are two, by using these two types of methods, the genes are synthesized. So these genes are synthesized. For this, there is no donor required. For these, there is no donor required. So these gene synthesis offers greater flexibility and control over the gene sequence, while reverse transcription is a more direct approach when messenger RNA for the gene is readily available. So these two different methods are helpful in the synthesis of genes artificially. Now, next question is DNA ligase and DNA polymerase are two enzymes that are used in genetic engineering. Complete table 8.1 to show the roles of DNA ligase and DNA polymerase in genetic engineering. So, DNA ligase and DNA polymerase. One enzyme is used. Role of genetic joins two sections of sugar phosphate backbone in DNA. So it is DNA ligase which acts as a glue. So it will act as a glue. So DNA for whereas DNA polymerase will not join the sugar phosphate backbone in the DNA. So adds a gene to a plasmid. Again adds a gene to a plasmid. Again, in genetic engineering, they are used. So it means they will act as a glue in the when there is phosphate backbone or sugar, two sections of sugar phosphate backbone in the DNA. And secondly, a gene when it is joined, when it is, you can say, added into the plasmid, the circular DNA. And the other type of enzyme, which is DNA polymerase. DNA polymerase is used when the nucleotides are joined together to make a polynucleotide. So adds activated DNA nucleotides to a polynucleotide. So here DNA ligase is not used, only DNA polymerase will be for this purpose. So in this way, these two different types of DNA molecules are there. Now we will move towards the next question. And next question is the polymerase chain reaction, PCR. Polymerase chain reaction, PCR, is used to make many copies of a gene. Three temperatures are used in PCR cycle. State the three temperatures that are used and outline what happens at each temperature during a PCR cycle. So we have to state the three temperatures that are used and outlined. So here, this is very simple question. This is very simple question. We have only add the different steps and the different temperatures. And first step is, I have written all this, and number one step is denaturation. Denaturation. And what happens? Denaturation occurs at high temperature. It occurs at high temperature. And high temperature means it can be 95 degrees centigrade, about 95 degrees centigrade. So what happens at high temperature? At high temperature will breaks down the, it will breaks down the hydrogen bonds. Bonds holding the DNA strands together. So this results the separation of DNA into two single strands. So in this way, you can see the DNA strands. These DNA strands are there. So the bond between them will break down. So these bonds will break down. As a result, there will be two strands of DNA. They will get separated from one another. And second, you can say step is annealing. Because two strands of DNA, they are joined together with the help of hydrogen bonds, which occur between adenine, the four nitrogen bases, adenine, thymine, guanine, cytosine, two bonds between adenine and thymine, and three triple bond between guanine and cytosine. So in this way, these bonds will break down, and as a result of breakdown of this bond due to denaturation, due to high temperature, these two strands of DNA will get separate from each other, and as a result of this, and the second is annealing, which takes place at low temperature. So low temperature is required for that, and that low temperature is about 50 to 65 degree centigrade. So what happens at 50 to 65 degrees centigrade? So this temperature is lower to allow the primers. What are primers? The short single strand DNA sequence to bind to the complementary sequence on the single-strand DNA templates. Primers are designed to flank the target DNA region that needs to be amplified. So after this third part is elongation or extension. Extension. Which needs intermediate temperature, that is about 72 degrees centigrade. So this is the optimal temperature for tag polymerase, the heat stable DNA polymerase used in PCR. So tag polymerase extends the primers by adding the free nucleotides to the three end of the each primer synthesizing new complementary DNA strands. So this results in the creation of two new double-stranded DNA. It will lead to the, so in this way, uh, these number one strand of DNA and number two strand of DNA from one. So in this way, so PCR polymerase reaction is a cyclic repetition. So these three steps, denaturation, annealing, and elongation, constitute one PCR cycle. This is the one PCR cycle. What happens after this? After typically the cycle is repeated for about 25 to 40 cycles in a thermal cycler, a machine that precisely controls the temperature changes with each cycle. So amount of target DNA sequence doubles, leading to exponential amplification. PCR utilizes temperature changes to control the denaturation, annealing, and elongation steps, allowing for the specific and efficient application of desired DNA sequence. And in this way, I would like to say that the different types of temperatures are involved for these three different types of steps. As a result of three different types of steps, you can say one DNA strand is, you can say, is changed into two. So this is a cyclic reaction. So this is called as polymerase chain reaction. Polymerase enzyme is used for over here. As a result of this, you can say a chain reaction is there. Cyclic reaction is there, which is about 25 to 40 cycles at a time. So it is, this cycle is repeated again and again. So it was all about the question number 7 and 8. And thanks for watching. Please like, share, and subscribe my channel. I have given my email ID in the description. You can ask me, you can question me. Thanks you very much.